Hello students. Today with regard to the topic that is plant anatomy, the last part that is the secondary growth. In earlier classes we covered the tissues and tissue systems. And this second growth undoubtedly it is a lengthy topic. And in this many key points are there and it is a concept based one. And keep the textbook with you. How my delivery is going to be synchronized with the text, we check it. And very first paragraph in case of the secondary growth, the apical meristem responsible for primary growth. The primary growth means plant grows in length. So primary plant body formed by apical meristems, what you call the primary growth. As plant increase in height, correspondingly there is an increase in weight. So primary plant body having primary permanent tissue unable to bear the weight of the plant. So there is a formation of the secondary tissues. As due to formation of the secondary tissue, there is increase in girth of the stem or root. So increasing girth of the stem or root, what do you call secondary growth? So secondary growth is due to, in order to bear this, what it is the weight, as a plant increase in height, secondary tissues are formed. And this secondary growth is present in most of the dicot, stem as well as root. In case of gymnosperm, stem as well as root, but is absent in case of monocot in very first paragraph. And this second growth in case of the steel, it is by vascular cambium, that is partly primary, partly secondary. And in case of the cortex, that is completely secondary cambium, that is cork cambium. So first, the intrastellar secondary growth, that is in case of the steel. And in case of the stem, collateral, conjoint, open, and dark vascular bundles present in a form of a ring. So ring-like vascular bundle arrangement is a characteristic feature of dicot stem. What we call it is UST. And within the vascular bundle, xylem inside, the phloem it is outside. And in between the primary xylem and primary phloem, a part of vascular cambium, that is nothing but intravascular cambium is present, that is completely primary. And medulla extended between vascular bundle, what do you call medullary ray. So adjoining cells of the medullary ray, that is parenchymatous medullary ray, adjoining to intravascular cambium, undergo dedifferentiation and produce strips of cambia, what do you call interfascicular cambia. So interfascicular cambium formed in between vascular bundle, it is secondary. Intravascular cambium present inside the vascular bundle in between the primary xylem and primary phloem is primary. Collectively, what do you call it is vascular cambium. So in overall, vascular cambium is a ring that is partly primary, partly secondary. That is formed in case of steel region. Next, the vascular cambium, it is active much inside than outside. And vascular cambium activity is influenced by environmental condition and physiological conditions of the plant. So as vascular cambium is much active inside, the more amount of the secondary xylem is formed inside. Whatever the cells cut off by this vascular cambium, inside transforms into secondary xylem, outside transforms into secondary phloem. Suppose it is vascular cambium, inside it is total, it is secondary xylem, outside it is total, it is secondary phloem. Suppose it is total, it is secondary xylem. The secondary xylem formed just below the vascular cambium is going to be the young, and far away it is the old. And youngest secondary phloem just outside the vascular cambium, and oldest secondary phloem nearer to primary phloem. So secondary xylem, secondary phloem located between primary xylem and primary phloem. So secondary vascular tissues are intercalated between primary xylem and primary phloem. And primary xylem more or less around the center. And secondary phloem and primary phloem are crushed due to more amount of secondary xylem. Why more amount of secondary xylem formed inside? Because vascular cambium is highly active towards inside. And these vascular cambium giving radiating rows of uni, bi or multiciliate rays, what do you call ray parenchyma. The ray parenchyma present in xylem, it is called xylem ray, phloem it is called phloem ray, collectively it is called vascular rays. So vascular rays formed by vascular chemium, which are secondary medullary rays. What are primary medullary rays? The extension of medulla between vascular bundle, what do you call? Primary medullary rays. And these are formed by vascular chemium, which are called secondary medullary rays. So primary medullary rays, secondary medullary rays. Primary medullary rays in primary plant body. Secondary medullary rays in secondary plant body formed by vascular chemium. So these are called vascular rays. Next, the vascular chemium activity depends upon the season. In spring season, it is highly active. It needs to be active because in spring season, new leaves are formed, new flowers are formed. So it requires more amount of water and mineral. So vessels, it should be with wider cavities. So many vessels with wider cavities are formed. The wood, what we call it is spring wood, or otherwise it is called early wood. As many vessels with wider cavities, what do you call it is less dense wood, lighter in color. So less dense wood, lighter in color, many vessels, wider cavities formed in spring season called spring wood or early wood. Next, in autumn season, plant shed off its leaves, require less amount of water and mineral. So vascular chemium is less active. It is not going to be inactive. It is going to be less active. It produces vessels with smaller cavities, few in number. So vessels, smaller cavities, few in number, which are much dense, dark in color, what do you call it is autumn wood or late wood. So spring wood, early wood, autumn wood, late wood. Spring wood and autumn wood together constitute one growth ring, otherwise what do you call annual ring. So annual rings or growth rings are formed by activity of vascular chemium, constituted by spring wood and autumn wood. The woods which are alternate are spring and autumn wood. And these growth rings are very well distinct in temperate and cold regions. Why? The there is no uniform climatic condition. There is a distinct seasonal variation, spring and autumn, like this. So we can expect the growth rings in case of the trees growing in areas like temperate and cold region. In case of the tropical areas, no distinct seasonal changes. So all which are uniform condition. Seasons are present, but seasonal variations are absent in case of tropical zone. That's why growth rings which are absent in case of the trees growing in case of tropical zones. There is one thing. You can estimate the age of the tree roughly by counting the number of annual rings. So suppose it is 100 annual ring. That means 200 woods are present. 100 it is spring wood and 100 it is the autumn wood. So we can estimate age of the tree by counting annual rings is called dendrochronology. It is not dendrology, it is dendrochronology. Dendrology is study of the trees. It is dendrochronology. Next. As the secondary xylem or wood, it loses power of conduction because the lumina of the vessel infiltrated with, filled with tannin, resin, aromatic substances, the gums, and other substances which become dark in color. Such a dark colored wood, what do you call it is heartwood. Because of aromatic substances, tannins and resin, it do not permit the entry of the microbes. So such a dark colored wood is commercially valuable, not useful for conduction. What do you call it is heartwood or duramen. So central wood, non-living, non-functional, provide mechanical support. The wood is called, that is heartwood or duramen. And recently formed wood around heartwood, light in color, what do you call it is sapwood. Otherwise what do you call arburnum. So in sap and heartwood, which wood is nearer to vascular cambium? Sapwood is nearer to vascular cambium. So sapwood which surrounds for it is heartwood. The sapwood gradually converted into heartwood. Again sapwood is going to be formed. Sapwood is with uniform thickness. So the wood with uniform thickness is sapwood, but diameter of the sapwood increases, but thickness remains same. But diameter and thickness of the hardwood, but it is increases. So sapwood is peripheral, light in color, not useful for conduction, not commercially valuable. Central, dark in color, commercially valuable, and not useful for conduction is heartwood or duramen. So alternate woods are spring wood and autumn wood. This is sapwood and heartwood. So annual rings are expected in sapwood and heartwood. Annual rings are found below vascular cambium. Next, due to intrastellar secondary growth, it exerts pressure on epidermis. It receives through cortex. So epidermal cells and cortical cells will be broken. So a protective layer has to be formed in case of the cortex. What do you call it? Is the periderm. So that is extrastellar secondary growth. That extrastellar secondary growth is initiated by a cambium called phellogen or otherwise called cork cambium. The cortical parenchyma cells undergo dedifferentiation and forms a couple of layer in thickness what we call phellogen. So phellogen it is formed by dedifferentiation from cortical parenchyma cell. It is a couple of layer in thickness, thin walled, nearly it is rectangular, and this phellogen or cork cambium gives more tissues outside, less inside. So outside it is the phellum or cork, inside it is secondary cortex or phelloderm. So phellogen or cork cambium, phellum or cork, phelloderm or secondary cortex, collectively what we call it is periderm. And the region exterior to the phellogen will die and slough off, and the cells, the cork cells which are suberized, it is impervious to water and gases. So exchange of gas affected in secondary cortex. So for exchange of gases in cork, bulged lens-shaped pores are formed called lenticels. So cork cambium, instead of giving cork at some places, it gives compact parenchymatous cells. What do you call it is complementary cells. The complementary cells located between cork cells given by cork cambium promote exchange of gases from atmosphere to secondary cortex. Because the complementary cells increase in number, epidermis got ruptured. So complementary cells promote exchange of gases from atmosphere to secondary cortex. So lenticels mainly concerned with gases exchange and also transpiration. Next. So whatever the tissues that occur outside the vascular cambium that constitute non-technically it is the bark. What are the tissues present in the bark? Secondary phloem, secondary cortex, phellogen and cork, which are all present in case of the bark. So what are the components of vascular cambium present in the bark? Secondary phloem. What are the components of vascular cambium absent in case of the bark? It is secondary xylem. So the bark formed in early season, that is soft, what do you call it is soft bark. In late season, the bark formed what do you call it is the hard bark.